Hello everybody. Welcome to the Antibody Society webcast series on antibody validation. In this, the 8th episode, we're going to discuss some methods for looking more broadly at antibody specificity and selectivity, protein arrays, and immunoprecipitation coupled to mass spectrometry. The Antibody Society is an international non-profit association. We're an international forum facilitating discussion, disseminating information, providing guidance, and assisting in training and advancement of students and postdoctoral fellows. So if you care about antibodies, please join the Society. The speakers in this webcast are experts from academia and industry. They're expressing their independent assessments and are not representatives of the Society. Viewers can write in questions at any time during the broadcast for the speakers to answer, and they will remain online for 15 minutes after the cast has ended to answer questions. The practical validation issues we've considered so far focused on whether an antibody is fit for purpose in a particular experimental context. Does it bind specifically to the antibody target of interest? This is an on-target validation, but it's only half the problem because of off-target binding. This includes the issue of selectivity, or does an antibody recognize an epitope specifically on other targets? But if we want to examine antibody binding in more depth, it can be useful to ask the more general question, what exactly can this antibody bind to? Arrays and immunoprecipitation coupled to mass spectrometry, IPMS for short, can give us a very detailed view of antibody specificity in a more global context. So in this episode, I'm very happy to be able to welcome Dr. Mike Taussig from Cambridge Protein Arrays and Dr. Fridtjof Lund-Johansen from Oslo University Hospital. Greetings, Mike. Greetings, Fridtjof. Thank you very much for joining us today. Mike, would you tell us about the use of protein arrays in antibody validation?
Hello, Simon. Thank you for the introduction. And first, before I start, I need to declare a commercial interest as CEO of Cambridge Protein Arrays Limited, a company offering protein array goods and services, in particular human proteome scale arrays. As related throughout this webinar series, the quality of antibody reagents is recognized as vital to getting correct and reproducible results. Validation of antibodies or other protein binders is therefore a problem of utmost concern for all those who are using antibodies as research and diagnostic reagents. This centers around the frequent reports that many research antibodies against proteins fail to do their job, either by cross-reacting with a different protein or even not recognizing the desired target at all. This has caused several instances of misleading or erroneous data entering the literature, especially with techniques like Western blot or immunohistochemistry. One way to confirm specificity for a protein target is to screen the antibody for binding to the widest possible number of proteins. And this is the principle behind protein arrays as validation tools. As shown on the right, it is now possible to present a vast number of proteins as individual spots on a glass microscope slide and react antibodies with them efficiently and economically. Protein array data can address essential questions around antibody specificity and selectivity, including whether the intended target is recognized, whether the antibody is monospecific or cross-reactive, and in that case, what the cross-reactions are with, how many there are, how strong, and how relevant to the proposed application. A cross-reaction may fade away at the working concentration of the antibody, or be irrelevant if the cross-reactant is rare, compartmentalized, or at low concentration. Protein arrays can also distinguish conformational and linear epitopes, and thus suitability for downstream applications. Illustrated is a block of a protein array with all the proteins revealed, and on the far right, those reactive with a particular antibody. The central red spots are duplicate reactions with the right target, but there are some other reactions at lower intensity and these may turn out to be significant cross-reactions. Essentially, protein arrays are highly multiplexed, solid-phased immunoassays, monitoring the reaction with hundreds or thousands of immobilized proteins in parallel. Through recombinant protein expression systems, the number of proteins now available is sufficient to cover most of the human proteome, and probably similarly for the mouse. Since one microscope slide can hold more than 40,000 spots, it provides the equivalent of 20,000 duplicate data points for a thorough analysis of specificity. Antibody consumption is low, and sensitivity is similar to an ELISA. On the far right is a full array of the type we use, a slide with over 20,000 proteins, and next to it, an expansion of one array block. This was stained against a tag on each protein with false color images reflecting the concentration of each spot, white being the highest, followed by red, etc. Antibody or other binder validation is an often requested use of protein arrays and can be used for all binder types. In addition, some other applications are listed here. The arrays are also ideal screens for autoimmune sera, where the antigen is still to be discovered, as well as other types of interaction, including protein-protein, protein nucleic acid, and protein small molecule binding, which, for instance, can reveal off-target reactions potentially critical to the use of a new drug. The arrays can also be used as substrates for protein-modifying enzymes such as kinases and glycosylases. I should also mention briefly peptide arrays, which can define the linear epitopes of both reagent and serum antibodies. I also want to note that the term protein array has sometimes been used to mean antibody array, which is not what I am going to cover here. Those use banks of immobilized antibodies to identify free proteins, for instance, as biomarkers, not the antibodies themselves. Like any technology with a degree of complexity, protein arrays present a number of variables. Most important is the quality of the proteins, which will be produced recombinantly by prokaryotic or eukaryotic systems or cell-free systems, which have become very versatile. The expression system will affect features like glycosylation or other modifications. One frequently voiced concern is whether the proteins are correctly folded. And where there is a large number of proteins with no particular assay method, this can be hard to demonstrate. Our own experience is that in many cases there is evidence of correct folding of soluble proteins, which can also be monitored by intentional denaturation. Integral membrane proteins, such as GPCRs, are in a particular category, of course, but this may not affect specific binding of an antibody to linear epitopes. The other issues listed include the type of surface favored, which will dictate the readout device, and which may be planar, as on glass slides or membranes, or on beads. The chemistry of attachment to the surface, either covalent or by adsorption. For slides, how the proteins are spotted and in what concentrations. Are they native, full length, fragmented or denatured? How binding is detected, often through a labeled secondary antibody or via a biotin, streptavidin or other tag. The configuration of the scanner for fluorescent readout, and then the procedures involved in analyzing the data, evaluating the results, and interpreting their significance. I will have to leave these technology aspects to some plentiful reviews on these topics. Now, this slide shows the steps involved in making a large-scale protein array, which has been expanded to near proteome size. It proceeded from a clone library, then gateway cloning of the open reading frames with fusion to a purification and detection tag, in this case GST, sizing, conformation of DNA, then large-scale protein analysis in yeast, in this case, The proteins were then individually purified on GSH columns, followed by SDS page for molecular weight confirmation, before final printing on the slide. Clearly, this is a lot of work for a dedicated team, so that commercial protein arrays are not cheap to produce. Though because of the economical printing and sensitive readout, a large batch can be produced from one protein production run. So now we get to how the arrays are used in antibody validation. And as shown, the principle is basically that of an ELISA, albeit a highly multiplexed one, where the antibody is incubated on the protein array slide, then washed to leave molecules bound to the target. Notice here that the proteins are spotted in duplicate, which is essential for a reliable readout, and then detection with a fluorophore labeled anti-Ig secondary. Shown is the specific binding and a weaker cross-reactive one, so that after the image is scanned, the binding can be visualized as a series of fluorescent spots. The array carries the identities of all the proteins, and it is necessary to fit these spots to a grid to determine which one is which. And this is partly done by grid fitting software and checking by eye. The intensities of all the proteins are then expressed in a data file from which various analyses can be started, for example, in R. It is important to subtract values attributable to the developing reagent. This will have its own fingerprint of reactivities, which must not be confused with that of the sample. To evaluate the significance of the binding data against hundreds or thousands of proteins, statistical methods are applied, of which a basic one is the Z-score, i.e. the number of standard deviations away from the population mean for any particular value in a normal distribution, which denotes the probability that the result could have arisen by chance. A Z-score above 3.5 is a useful one to apply and potentially meaningful. But for the most specific interactions, we expect Z-scores above 5, where the p-values become very small. Since not all proteins are spotted at the same concentration, an adjustment is made based on protein level, determined at the same time in a separate fluorophore channel, for example, by an anti-tag antibody. Once the fluorescence intensity results have been acquired with all the correct parameters, the proteins recognized, or the hits, can be identified and the specificity of the antibody evaluated. Here are three quasi-real-life examples with the fluorescence intensity on the y-axis, and the proteins identified on the x. The scanned image around the protein of interest is shown on the left. AB1, antibody 1, is a highly specific antibody with very limited cross-binding. Incidentally, the target indicated in gray is actually a known Fc receptor. Antibody 2 is quite cross-reactive, with some proteins exceeding the target in intensity. Target is in orange. And finally, antibody 3 is highly cross-reactive, sticking to many proteins, of which the true target is only one of medium strength. And essentially, this would be useless as a reagent. Now, I mentioned earlier that protein arrays can also indicate fitness for applications requiring either the correct protein conformation or after denaturation, which can be done by comparing a native array with a denatured one, after treating the whole array with a denaturant such as 8 molar urea and DTT, or by heating, imitating techniques such as Western blot or immunohistochemistry. This example is of two monoclonal antibodies, of which one, the anti-SMAD4, reacts solely with the denatured array, probably with a linear epitope revealed by denaturation, while the other, anti-ACO2, binds to a conformational epitope which is lost on denaturation. For a thorough analysis of an antibody, it can be useful to assay on both types of array, native and denatured. Finally, to summarize the advantages of validating antibody specificity on protein arrays, we can point to the ability to screen against a vast library of proteins at the same time, which may either confirm monoreactive target specificity or demonstrate cross reactions and off-target binding, and identify what they are. Linear and conformational epitopes can be distinguished. The data can give the user confidence that their antibody is fit for purpose, at least from the point of view of specificity, or alternatively, it can be a warning that it should be used with caution, both for reagents and therapeutics. It's always best to go into antibody applications with eyes open, and the arrays can be an important aid in so doing. Any questions?
Thank you, Mike. I guess whatever the technology we use, we're always concerned about how realistic the target molecules are compared to what we find in the experimental context. It's a beautiful technology you describe, and I know you mentioned these issues in your talk, but would you like to comment on how you assess whether the protein arrays, the proteins on the arrays are folded in a native conformation?
Well, thank you for that question. One point I should make is that while the aim is to have full-length proteins on the array, this applies to individual chains, and the arrayed proteins are single chains, but not multi-component proteins. So if you have a protein which has two or more chains which need to fold together, they are not expressed on the array. We have the individual chains. So if the conformation depends on having more than one protein chain there, then that will obviously limit the use of that array for that particular protein. However, we're looking at antibody validation, and it's likely that the epitopes will be present on the individual chains as well, in most cases. As I pointed out, if you're looking for specific conformations of 20,000 proteins, but you know very little about the activities of these proteins, it's hard to know what criteria to use. However, we have many examples by now of antibodies which are dependent on protein conformation and which will react with the slides. We will react with the arrayed proteins. And therefore, we have a pretty confident feeling that many, most, if not perhaps all of the proteins are in fact properly folded. Now, a particular problem arises with membrane proteins which require the lipid environment for their folding. There, one might be able to determine specificity of linear epitopes, or one might have to, for confirmation, turn to arrays which are coming on the market in which the proteins are expressed in a lipid environment to maintain their folded form.
Thank you, Mike. That's a very informative answer. Thank you. Of course, we should point out that there are also commercial sources for arrays of peptides to investigate epitope specificity, and also, as you say, specifically for human membrane proteins now. Arrays display a wide and can display an almost proteome-wide range of defined targets. So that can really help us define how strongly an antibody binds to off-target as well as on-target proteins and peptide sequences. Very useful technology. But of course, arrays define in advance precisely what they display for the antibody to bind. So how can we look in a more general manner for targets that an antibody may bind to? IPMS is a technique for examining binding specificity and selectivity in depth, and it may solve many of the problems we have encountered during this series. So it's a pleasure to be able to welcome Dr. Fridtjof Lund-Johansen from the Department of Immunology at Oslo University Hospital to describe the advantages of this and related technologies using mass spectrometry. Greetings, Fridtjof.
Thank you, Simon. In this presentation, I will first go through some of the more commonly used methods to test the performance of antibodies and validate them. And then in the second half, I will explain how antibody array analysis can be combined with immunoprecipitation and mass spectrometry to meet needs for both high throughput and high precision. In 2016, an international working group on antibody validation published a paper in Nature Methods where they described 5 methods, or pillars as they call them, for testing antibody specificity. Even though the paper did not really contain anything conceptually new, I think that it was very important because it has been used as a starting point for discussions. And today, the paper has as many as 188 citations in Google Scholar. Among the methods were knockout, correlation, overexpression, and immunoprecipitation-mass spectrometry. I will go through each one of these and point to strengths and weaknesses, and I will also speak about protein arrays, which were not included among the pillars, but which have been reported more lately to be very good tools to identify monospecific antibodies. First of all, knockout. I think there is consensus among scientists and also in the industry that knockout is the gold standard. Knockout means that you target the disruption of DNA encoding the intended target, so you obtain very, very clear negative controls. On this Western blot from the AbCam catalog, you can see in the left lane protein from wild-type HAP cells stained with an antibody, and the antibody sees a strong predominant band. And this band is gone in the knockout sample. And most people will agree that this is very strong evidence that the predominant activity of this antibody in HAP cells is the intended target. However, the blot also shows a challenge with knockout testing, and that knockout testing is very often done in one cell type. But those who work with Western blotting know that extra bands are quite often sample dependent. So in this blot, you can see 2 strong bands that appear in protein from Raji cells, where they're seen most clearly, and they're not easily visible in the lanes from the HAP cells. So to test specificity, you need to both know on-target binding and off-target binding. And since off-target binding can often be sample dependent, the best way to assess it is to test the antibody for reactivity against a large number of different samples. This is what Bio-Rad did for this antibody, where they tested reactivity of the antibody against a large panel of cell lines. And as you can see here, in all the cell lines, there is a single predominant band. And this means that the antibody is monospecific consistently in all these cell types. Correlation is perhaps a little bit more difficult to deal with. I think it is analogous to fingerprint matching. And the concept is that the staining pattern of an antibody across a large series of samples is analogous to a fingerprint. However, to test the performance of this type of validation, you really need to have a fingerprint database to determine how good the match is from your print to the prints in the database and how many matches there are. Ideally, you'd like there to be only one. So how can this be done? And I think this was not described very well in the paper from 2016. What you can do is that you can analyze published data sets from mass spectrometry. With mass spectrometry, you can detect thousands of proteins, and data sets with proteins from human cell lines are quite useful to determine how unique protein expression profiles are. What I've done here for this line plot is to determine the frequency of matches at any level of correlation between 0.5 and 1 in data sets from between 6 and 11 cell lines. At a correlation of 0.5, you will find 1,000 matches or more for any given protein expression profile. And even at a correlation of 1, which is perfect correlation or identical profiles, you'll very often find that there are many matches. The implication of this is that any staining pattern, even across 12 cell lines, is not going to be completely unique. So you cannot get definitive validation by this approach, at least not yet. My hope is that with even more complex samples, you probably can, but we will see this in the future. Overexpression is something that, in my opinion, is a last resort when everything else fails. Typically, the manufacturer has tested the antibody on all the cell types they usually work with and probably not found something that looks like a convincing result. So what you can then do is that you can overexpress the intended target in one of the cell lines, and then, of course, you will most likely see something that looks like convincing staining. This approach was evaluated in detail in a study published in 2014. The authors tested 1,544 antibodies that failed to detect anything convincing on Western blots from wild-type cell lines. They then prepared cell lysates with overexpressed proteins, and 82% of the antibodies now seem to work very well. Unless you can be sure that the wild-type cells really did not express these targets, I would say that this type of evidence is really evidence that the antibody is not performing very well, since it is probably not able to detect endogenously expressed protein. And usually that's what you want to do with the antibody. Immunoprecipitation-mass spectrometry, or IPMS, has been suggested to be a gold standard for antibody validation, since this assay can yield sequence-based information about antibody-bound proteins, and that is as direct as it gets. A paper in Nature Methods evaluated this approach in detail. They tested hundreds of antibodies, and the antibodies were sent to different laboratories to test inter-laboratory reproducibility. The conclusion was that for antibodies that were specific, the intended target typically was found among the top 3 most abundant proteins in the immunoprecipitate. The challenge with this approach is that usually the immunoprecipitate contains hundreds of proteins, and it's difficult to determine what of these 100 proteins are interaction partners of the intended target, which ones are cross-reactive proteins, and which ones are simply sticking to the beads used for the pull down. So IPMS, when used in this way, which is to use antibodies to pull down proteins from total cell lysates, can give very strong evidence for on-target binding, but it can be difficult to interpret off-target binding. Protein arrays should in principle be the ideal tool to assess antibody specificity, because you can probe an antibody against the entire proteome in one assay. Yet, protein arrays are controversial. In a study published in 2018 in Nature Methods, protein arrays were used to identify antibodies in primary screening that were monospecific for human transcription factors. The antibodies were then tested in immunoprecipitation and Western blotting. And the conclusion was that most of the antibodies that were monospecific on the primary screening on the arrays were also good in immunoprecipitation and Western blotting. Now, the limitation of this study is that the secondary testing was done using lysates with overexpressed proteins. And as I explained earlier, this is not a very stringent test for antibody performance. And the implication is that we don't really know yet how good protein arrays really are. So to summarize all this, I would say that the consensus today is probably that knockout testing is the gold standard and ideally the way every antibody should be tested. The problem, though, is that knockout testing is slow and expensive. So can we envision that large numbers of the antibodies on the market will be tested by knockout testing in foreseeable future? I would say the answer to that is probably no. So the challenge of validation is not really only one of precision, but it's also one of throughput, because there are more than 2 million antibodies on the market. And if we want to identify the best ones, how do we screen through the whole haystack? In our laboratory, we have developed technology to deal with just this. We have developed array technology so that we can work with thousands of antibodies in parallel. Our arrays consist of latex beads with fluorescent barcodes. We can make as many as 4,600 different beads using 2 sizes and 5 fluorescent dyes, so we can work with thousands of antibodies in parallel. When we make antibody arrays, the antibodies are coupled to the color-coded microspheres, and they are used to immunoprecipitate biotinylated protein from samples such as cell lysates. To detect protein binding, the beads are stained with streptavidin, which binds to biotin, and the streptavidin is fluorescent by phycoerythrin. And then the beads are analyzed in a flow cytometer capable of reading the color codes from the beads and also measuring fluorescence from streptavidin. We also use these beads to make protein arrays. In this case, the beads are coupled with an antibody to an anti-tag antibody, GST, and we have as many as 12,000 recombinant human proteins with a GST tag. A year ago, we published a method analogous to a capture Western blot or an antibody array Western blot. The sample preparation is very similar to what is used in standard Western blotting. So proteins from cell lysates are denatured by heating with SDS and reduced, and they are separated by gel electrophoresis. However, the gel electrophoresis that we use yields the proteins as insoluble fractions. So it's preparative gel electrophoresis using an instrument called gel-free. What we obtain is a micro plate where each row is analogous to a lane on a Western blot, and each well contains proteins with a narrow size distribution range. As an example, this cartoon to the right, which shows an antibody that binds a protein that elutes in fraction 5 and 6 due to its size, and the bead with this particular color code is then used to immunoprecipitate that protein. Among thousands of beads, we can then make one line chart for this particular one showing a peak in fraction 5 and 6. And this peak is then analogous to a band on a Western blot. An important difference between Western blotting and antibody array analysis is that the antibody array analysis and flow cytometry yields data in a numerical format. So we can not only work with thousands of antibodies at the same time, but the results we get are also qualitatively different because we obtain numbers. And in the next slide, I'll show you how this can be used. The heat map you see here is really a spreadsheet with numbers with 3,706 antibodies. And we show reactivity patterns across 12 fractions from each of 6 cell lines. What we then did is that we did cluster analysis to identify antibodies with similar reactivity patterns. The heat map to the right is an enlarged section of the little yellow box in the left heat map. And as you can see here, antibodies to AKT1 cluster together and away from antibodies to CRAF2, even though both types of antibodies bind 55 kilodalton proteins that are broadly expressed. So what this shows is that antibody reactivity patterns that are only slightly different can be recognized as according to their specificity using computer algorithms. Yet, even though the correlation between these reactivity profiles is as high as 0.95, the results are not yet definitive because there's usually a couple or maybe as many as 10 other proteins that have exactly the same distribution profile in these fractions. So the approach is very, very good to identify good candidates for what we can call definitive validation. And in the next slide, I'll show you how that is done. We don't use all of the fractions or the entire material for the antibody array analysis. We only use an aliquot of each, so we have material left to work with. Now that we have data from antibody array analysis, we can go back to those fractions and identify fractions that are enriched for the targets of specific antibodies. In this case, an antibody to the retinoblastoma protein, I then bound strongly a protein that eluted in fraction 5 from U2OS cells. So we take this fraction and use it for immunoprecipitation with that antibody. And now we trypsinize that immunoprecipitate and analyze the peptides by mass spectrometry. On the bar chart to the right, you see one blue bar, and this bar corresponds to proteins from the sample. Now, how do we know that? We know that because the cells were first cultured in the presence of isotopes that incorporate into all the proteins. And these isotopes can be recognized by the mass spectrometer and as different from all the black bars that contain isotope negative proteins. So the isotope negative proteins are typically keratins and immunoglobulins and blocking reagents, but retinoblastoma protein is the only protein in this IP that derives from the sample. So with this approach, we get IPMS data that are much simpler to interpret. We do not have 200 proteins or more. We have one or usually less than 10. And in most cases, at least 90% of the signal comes from one target. So the data are much simpler to interpret. And now we can use IPMS for specific definitive specificity assessment and also then determine potential off-target binding. So far, I've been talking about denatured proteins and Western blotting applications, but of course, antibodies are used for a wide number of applications, and some of those are detection of native proteins. To detect native proteins, we use fractionation methods for native proteins, such as subcellular fractionation and size exclusion chromatography, which is used to separate native proteins in context of their interaction partners. It's used quite a lot to work with protein complexes. And this cartoon here shows example of an elution profile of targets of an antibody that recognizes a monomer, which is the peak to the right, and then the same protein found in the complex, which will elute earlier. This slide shows what the real data look like. These are line charts, and each line corresponds to a different subcellular fraction. The line chart to the left shows the elution profile of targets of an antibody to REL-A, which is a protein found in a large complex in the cytosol, corresponding to this blue line. PDIA3 is a protein found inside of cytoplasmic organelles, corresponding to the red line here, whereas cyclin B1 was found in a fraction enriched for both cytoskeleton and soluble nuclear proteins. Now that we know what antibodies bind proteins in what fractions, we can now go back and do immunoprecipitation again and mass spectrometry. This is an example of a heat map showing chromatograms for the targets of hundreds of antibodies, and we can in principle do thousands of antibodies. The advantage of array-based analysis is that it's very fast. So you can work with very large number of antibodies. And now you can go to the fractions and do your immunoprecipitation from enriched fractions, such as we did in this case. We used an antibody that bound a target in the membrane fraction here, in fraction 13, immunoprecipitated with that antibody, trypsinized the immunoprecipitate, and analyzed the peptides by mass spectrometry to find that this antibody binds a protein called OXCT1. Protein arrays in our hands are very useful tools, but the results are not always what you might expect. What we typically see is that the intended target is on the top 5 list of binding proteins. However, we very often see cross-reactivity on these arrays, and we do not necessarily find the same cross-reactivity in applications such as Western blotting or immunoprecipitation. So we think that protein arrays are good to obtain evidence for on-target binding, but it's not necessarily easy to interpret the cross-reactivity, which may not be very predictive of what you will obtain in other applications. So to summarize this, we think that antibody validation is a question of both precision and throughput. And with arrays, you can obtain very high throughput. But to really benefit from that, you need to separate proteins in samples. So you need Western blotting applications, or the native versions of it, which is a combination of subcellular fractionation and size exclusion chromatography. But if you combine very extensive protein separation and antibody array analysis, you can get very high resolution. And it's easy to work with large numbers of fractions when you have bead-based arrays, because you can use liquid handling robotics and automated flow cytometry. So you can go through a very, very large number of antibodies and large numbers of fractions and go back to your data and then identify fractions that are enriched for the targets of individual antibodies. Now you can immunoprecipitate from those fractions and do mass spectrometry to get the high resolution specificity assessment. And this is what I really wanted to explain to you today. Thank you.
Thank you very much indeed, Fridtjof. So as long as it's possible to get the protein into solution in a non-aggregated form, the technology is applicable to pretty much any target. But aren't there a number of issues in comparing quantifications derived from mass spectrometry measurements, or is that just old hat these days?
I think that mass spectrometry, as we use it, this is so label-free mass spectrometry, is semi-quantitative, and as it probably is just as good as fluorescence analysis. I mean, in flow cytometry, you typically have a dynamic range of anywhere between 3 and 5 decades. With mass spectrometry, the dynamic range is probably higher than that, and up to 7 decades. So I don't think that quantitation is the issue here.
Great, thank you. So if you want a full definition of what an antibody can bind to, immunoprecipitation-mass spectrometry is hard to beat. But of course, it can't deal with the various other experimental contexts that we've discussed in the series, like immunohistochemistry or Western blot, for example, because it's only applicable for antibody binding in solution. Thank you very much indeed, Fridtjof. Over the past few episodes, we've seen that a combination of genetic knockouts, arrays, and IPMS can go to almost any desired depth to investigate exactly what a particular antibody can and can't bind, and so very robustly interpret and validate data we get from standard techniques like Western blot and IHC. Of course, in this series, we've pointed to major issues of antibody validation, but we've avoided many big topics, such as validating antibodies against post-translational modifications, but that will be a series for itself. But we hope that users will now have a better feel for the challenges that they face. In the final 2 episodes, Andrew Bradbury, and Andreas Plugtun will look into the future of affinity reagents. We hope you will join us with them. This is the Antibody Society webcast series on antibody validation. Thanks for joining us, and bye-bye till next time. The series would be impossible without the support of our many corporate sponsors. Mike Taussig and Fridtjof Lund-Johansen will be online for the next 15 minutes and will be very happy to answer any questions you may have. If you have any questions on the use of array technologies and IPMS for validating antibodies for Mike and Fridtjof, please type them in at the question and answers tab now.